Welcome to G Fun Facts Online. Today we are embarking on a journey inward, exploring the most complex structure in the known cosmos. It is a 3-pound universe of gelatinous tissue that you are carrying around inside your skull right now. For centuries, explorers have sought to map its terrain, not with ships and compasses, but with scalpels, microscopes, and eventually supercomputers. This is the story of neuromapping and brain atlases, the centuries-long quest to draw a map of the human mind. From the crude sketches of phrenologists to the digital Google Earth of the brain being built today, this field represents the intersection of anatomy, technology, and philosophy. It is a discipline where a difference of a few millimeters can mean the difference between moving your hand and being paralyzed, between speaking fluently and losing your voice forever. So let us strip away the skull and look at the history, the technology, and the future of how we navigate the human brain. To understand where we are today, we have to understand where we started. The desire to localize function in the brain is ancient, but the scientific journey truly began with a misstep, a famous mistake called phrenology. In the early 19th century, a man named Franz Joseph Gall proposed that the brain was divided into 27 distinct organs. He believed each of these organs was responsible for a specific personality trait, things like benevolence, hope, or destructiveness. Gall believed that if you had a lot of benevolence, that part of your brain would be larger, creating a bump on your skull. Now, while phrenology was scientifically flawed and eventually dismissed as pseudoscience, it planted a crucial seed. It introduced the idea of functional localization, the concept that specific parts of the brain do specific things. We moved from this pseudoscience to hard evidence through tragedy. One of the most famous stories in neuroscience is that of Phineas Gage. In 1848, Gage was a railroad foreman who had a tamping iron blasted through his left frontal lobe in an explosion. Miraculously, he survived. But the polite, hard-working man his friends knew was gone. He became fitful, irreverent, and impulsive. This tragic case study provided the first hard evidence that the frontal lobes were the seat of personality and social behavior. It wasn't just a gray blob. Different parts held different aspects of who we are. Soon after Gage, in 1861, the French physician Paul Broca met a patient named Louis Victor Leborgne. This patient could understand language perfectly well, but he could only say one syllable, tan. When the patient died, Broca autopsied his brain and found a specific lesion in the left frontal lobe. This area, now known as Broca's area, was the first anatomical proof of a language center in the brain. If that area is damaged, you lose the ability to produce speech. But the true Mercator projection of the brain, the map that has stood the test of time, arrived in 1909. A German anatomist named Corbinian Brodmann spent years hunched over a microscope analyzing the cellular structure, or cytoarchitecture, of brain slices. He noticed that the cortex, the brain's outer layer, was not uniform. In some places, the layers of cells were thick and dense. In others, they were thin and sparse. Brodmann divided the cortex into 52 distinct areas based on these cellular patterns. Remarkably, his map was not just anatomical, it turned out to be functional. For example, Brodmann Area 4 is the primary motor cortex, controlling voluntary movement. Area 17 is the primary visual cortex, processing what you see. Even today, more than 100 years later, neurosurgeons and researchers still refer to Brodmann areas as the standard coordinates of the cortex. It remains the most enduring map in neuroscience history. Now, let us step into the modern era. Modern cartographers of the mind do not use quill pens or simple microscopes. They use magnetic fields and quantum physics. The revolution in brain mapping is driven by imaging technology that allows us to peer inside the living skull without making a single cut. The foundation of this is magnetic resonance imaging, or MRI. MRI uses a massive magnet, often 1.5 to 7 Tesla in strength, to align the protons in the water molecules of your body. When a radio frequency pulse knocks these protons out of alignment, they emit a signal as they spin back into place. Since different tissues, like gray matter, white matter, and fluid, have different water contents, they emit different signals. The result is a high-resolution, 3D structural map of the brain's anatomy. An MRI scan is like a satellite photo of a city. It shows you the streets, the buildings, and the parks. It is a beautiful structural canvas. However, a structural map doesn't tell you where the traffic is flowing or what the people inside the buildings are doing. For that, we need functional MRI or fMRI. To see the traffic or neural activity, scientists rely on the BOLD signal, which stands for blood oxygen level dependent. Here is how it works. When a specific brain area is active, say, when you tap your finger or solve a math problem, it consumes more oxygen. The body rushes oxygenated blood to that spot to fuel the activity. Oxygenated blood has different magnetic properties than deoxygenated blood, and the fMRI scanner can detect this subtle change. If an MRI is a map of the city, fMRI is the traffic report. It lights up the roads that are busy during specific tasks, allowing researchers to create functional maps for language, memory, and emotion. But there is another layer to this map. The brain's gray matter, the computing centers, is connected by white matter, the cables. To see these cables, we use diffusion tensor imaging, or DTI. This technique tracks the movement of water molecules. In the brain's white matter tracts, water flows more easily along the nerve fibers than across them, much like water flowing through a hose. By tracking this directional flow, DTI allows scientists to reconstruct the brain's wiring diagram. This process, called tractography, produces stunning, rainbow-colored images, where red might represent fibers running left to right, blue for up and down, and green for front to back. It reveals the superhighways of information transfer, such as the corpus callosum connecting the two hemispheres and the arcuate fasciculus connecting language areas. With these tools, we can take pictures, but a map is useless without a coordinate system. If a surgeon wants to target a tumor, they need a precise reference frame. This led to the creation of standardized brain atlases. For decades, the gold standard was the Tallefix and Tourneau Atlas from 1988. It was based on the post-mortem dissection of a single brain, that of a 60-year-old French woman. Researchers created a 3D coordinate grid based on this one brain, and for years, every other brain scan was warped and stretched to fit this grid. However, relying on a single elderly brain had major drawbacks. It didn't account for the massive variability between individuals. A young man's brain and an elderly woman's brain can differ significantly in shape and size. The scientific community realized they needed a population-based map. Enter the Montreal Neurological Institute, or MNI. Researchers there scanned hundreds of young, healthy brains and used supercomputers to average them together. The result, known as the MNI 152 template, is a blurry, idealized brain that represents the average human anatomy. Today, most neuroimaging software automatically registers individual brains to the MNI template. It is the Greenwich Mean Time of neuroscience. If a research paper says a specific activation was found at coordinate 34, -12, 50, they are almost certainly referring to MNI space. But even an average has its limits. The most advanced frontier in atlasing comes from the Jülich Research Center in Germany. The Jülich Brain Atlas is a probabilistic 3D atlas that goes far beyond the MNI template. It is based on the microscopic analysis of 10 distinct post-mortem brains. Because every person's brain is folded differently, a specific area like Broca's area might be a few millimeters to the left or right in different people. The Jülich Atlas captures this variability. Instead of drawing a hard line, it gives a probability. It might say, there is a 90% chance this voxel is part of the visual cortex, and a 10% chance it is part of the association cortex. It is the first map to truly account for the biological diversity of the human mind, acting like the Google Earth of neuroscience. We have moved from mapping regions to mapping connections. This is the era of connectomics, the attempt to build a comprehensive wiring diagram of the entire nervous system. Launched in 2010, the Human Connectome Project, or HCP, is effectively the Human Genome Project of neuroscience. Its goal was to map the macroscopic connections in the healthy human brain. Using custom-built MRI scanners with ultra-high magnetic gradients, the HCP scanned 1-200 healthy adults, including twins, to understand how brain wiring relates to genetics and behavior. The HCP produced a new parcellation of the cortex, identifying 180 distinct areas per hemisphere, 97 of which were previously unknown. They found that the brain is not just a collection of blobs, but a mosaic of highly specialized tiles, each with its own unique fingerprint of connectivity, function, and structure. However, mapping the brain at a cellular level generates data on a scale that boggles the mind. This is the petabyte challenge. A single cubic millimeter of brain tissue, about the size of a grain of sand, contains 50,000 neurons and 130 million synapses. Imaging this tiny speck at nanometer resolution produces 2 petabytes of data. To map a whole human brain at this resolution would require zettabytes of storage, more than all the digital content in the world today. This big data challenge has turned neuroscience into a computational science. Researchers now use AI and machine learning algorithms to automate the tracing of neurons, a task that would take a human thousands of years to do manually. Despite our advanced maps, vast territories of the brain remain terra incognita, or unknown land. Deep beneath the cortex lies a thin, sheet-like structure called the claustrum. It is one of the most densely connected regions in the brain, receiving input from almost all cortical areas. Francis Crick, the co-discoverer of DNA, and Christoph Koch famously hypothesized that the claustrum acts as the conductor of the brain, integrating distinct sensory inputs like sight, sound, and touch into a single unified conscious experience. Because it is so thin and irregular, it is notoriously difficult to map with standard MRI, making it one of the final frontiers in structural mapping. Similarly, most atlases focus on the cerebral cortex, the wrinkled surface, because it is large and easy to image. However, the subcortex and brainstem are evolutionarily older structures controlling breathing, heart rate, and basic arousal. They are small, dense, and buried deep within the skull, where MRI signals are often distorted. Precision mapping of these life support systems is a major focus of current research, particularly for understanding comas and vegetative states. Now, why does all this mapping matter? These maps are not just for textbooks. They are used daily in operating rooms to save lives. Before removing a brain tumor, a surgeon must know exactly what the surrounding tissue does. If a tumor is located near the motor cortex or speech centers, what we call eloquent cortex, a wrong cut could leave the patient paralyzed or mute. Surgeons use neuronavigation systems, essentially GPS for the brain, that overlay the patient's MRI and DTI scans onto their real-world view of the open skull. They can see the invisible white matter tracks on a screen and navigate around them, preserving the patient's function while removing the cancer. Another incredible application is Deep Brain Stimulation, or DBS. For patients with Parkinson's disease, tremors are caused by misfiring circuits deep in the basal ganglia. DBS involves implanting a thin electrode into a specific millimeter-sized target, such as the subthalamic nucleus. Using high-precision atlases, surgeons can thread a needle through the brain to hit this tiny target with sub-millimeter accuracy. When the device is turned on, the tremors often vanish instantly. It is a miracle made possible by precise anatomical mapping. Looking forward, the future of neuromapping is simulation. Projects like the virtual brain are using atlas data to create personalized digital twins of patient brains. Doctors could one day simulate a surgery or a drug treatment on your digital twin to predict the outcome before ever touching your real brain. This computational modeling could revolutionize the treatment of epilepsy by predicting exactly where seizures originate and how to stop them. Furthermore, companies like Neuralink are pushing the boundaries of mapping by implanting thousands of microscopic electrodes directly into the cortex. To make these brain-computer interfaces work, we need maps that are not just static, but dynamic, capable of decoding the language of neural firing in real time. As our maps improve, the line between mind and machine blurs. High-resolution functional mapping is the key to restoring sight to the blind, movement to the paralyzed, and potentially allowing for direct brain-to-brain communication. In conclusion, the map of the human brain is far from complete. We have sketched the continents and named the major cities, but the winding backstreets and the private interiors of the houses, the neurons and synapses, remain largely unexplored. Neuromapping is the greatest cartographic challenge in human history. It is a journey inward to understand the very organ that allows us to understand anything at all. As our atlases grow more detailed, we do not just see the brain more clearly, we see ourselves. The map is the territory, and the territory is us. Thank you for listening to Fun Facts Online, and keep exploring.